Thank you, Leah. I'll just kick it off. If any of the committee members have questions, please turn your name tag. In your perspective, from your perspective, is it possible for all research laboratories to be Clia certified? Wendy, you've spoken to the increased expense of that. And if not, which I'm assuming would be the conclusion, where do you draw the line and what should or should not be returned to patients? What's the risk, or research participants? They're not necessarily patients. And what are the risks and benefits to those participants?
So to me, when you're talking about research laboratories, there's an enormous range in terms of research laboratories, and there's just no way that all research laboratories will be Clia certified. What I can imagine is if you try and think about what results would you want to be returned, should be returned, could be actionable and useful to return, is there a common, that's not all research laboratories, those are probably a subset of laboratories and probably a subset of technologies that are being used to generate those data. And so I guess, number one, no, I don't think every, R01 funded investigator should be necessarily making their laboratory into a Clia environment. There's just, as we were looking at with Carolyn's, there's an enormous amount of checks and balances and infrastructure for QA, QC that has to be put in place for Clia and CLAP certification, and that's just not a good use of resources, in my opinion. On the other hand, is it useful to have a certain number of laboratories that might serve as cores that could be either institutionally based or regionally based or nationally based that should be or could be Clia certified? I think that makes more sense to me in terms of thinking about this. And one could think about sending samples to a centralized place that at least is doing a lot of the, whether it's exome or genome or transcriptomics or things like that, but they're kind of a pipeline that's consistently used. That makes more sense to me. That's.
I don't think it's feasible or necessary to have all research labs be Clia certified, especially the ones on the discovery level. I think it's much more important to focus on, someone mentioned earlier that we have a reproducibility problem in biomedical science, and that is absolutely true, and the extent of it is not known yet. There are two efforts to try and figure this out, one within the government and one in the private sector by open science, but it's looking like only about 30% of all published biomedical research is reproducible, or less, which is even scarier. So if research is using human specimens, and this was, I was greatly disappointed to see that the comments that came forward from the NIH in their reproducibility workshop mentioned cell lines and all kinds of other things that are resources for research, but did not mention human biospecimens at all. So this is clearly a source, in my mind, of irreproducibility, and I mentioned the pre-analytical variables can make two seemingly identical specimens totally different in terms of their molecular quality and molecular composition. And there's no way you could reproduce them, especially if you did not know what happened to them, if you did not know their provenance. So I think that that reproducibility in terms of the analytes used and validation of the research, and this is not common in biomedical research either, that researchers either validate their own research, and they'll constantly tell you they don't have enough money to do that, and that's probably true, let alone have another outside lab validate their research, give them the recipe, and see if they can get the same answer. And certainly if they're not starting with the same analytes, they will never get the right answer. So I think that Clia certification is one level of assurance that you're doing things right in the right environment for rigor and reproducibility. But that will not, in fact, fix this problem of quality of analytes, fit for purpose, reproducibility on any level in research, especially if human biospecimens are your analyte of choice.
I have Greg, and then Chester, and then Wiley.
So I want to do a little thought experiment and get you all to sort of do a rapid-fire response. The normative decision has been made. Fran Visco and HIV groups have gone up to Capitol Hill, and now there's a decision that that there's going to be much more extensive data sharing with patients up front. And the committee, doesn't matter what this committee says, doesn't matter what we say, it's already been done. There's been questions on the committee about the cost and the other obstacles, but what are the things that need to be done up front if we move towards a vision of more expansive data sharing in the do we need legislative changes? Do we need policy changes? Do we need professional changes? If you had to name one or two things that we should think about right up front if the decision is taken out of our hands, what do you do?
I'll start. Policy changes, simplify the legal landscape of returning results, and then I think we have to think about how to protect trial integrity. So the timing of when results are returned, so as to not bias a clinical trial, for instance, where you're returning results and people start to say, well, maybe I'm on treatment or off a treatment, and therefore I'm on drug or placebo, and therefore I'm going to stop coming to the study because I'm seeing some results or not seeing results. So I think timing is really important about this as well.
I would say that, at least for the issue that I've brought up, you always want results going back to a patient on any level to be correct. That's to be true. And in order to assure that they are true, we need this base of control and standardization for the way specimens are processed and handled. And I think that it's more CMS in its payment for performance on the part of physicians that would control this and set up standards to do that through their professional organizations, then it would be Clia that controls what goes on in the laboratory. So it's CMS, yes, but it's also, it's payment for this very important, this is an investment upfront in the quality of the sample, which is an investment in the quality of the data that is going to go back to the patient. So we really need and you can't go back and fix it. Once it's fixed in formalin, that's it. So we really have one chance. It's upfront, it's immediate, it's time-consuming, labor-intensive, and it needs to be reimbursed. And it isn't right now. So I think that that's one thing that could be done.
So I think if the mandate is that we're going to return results, I think the realization in terms of as people are putting in grants and as NIH and other funders have policy in this is to adopt those same policies, but to allow for that to be done and done correctly. So I think in terms of budgets, it shouldn't be unrealistic to be able to build in that additional cost. And to the extent that there's infrastructure, whether it's NIH supported or however the community decides to do this, but infrastructure to allow to do this on scale. What I look forward towards, and we'll see how much all of us actually sets a standard for this, at least in genomic medicine, is there a standard that then gets set, or is there the infrastructure, whether it be in terms of the laboratories in which the data are generated, how the data are interpreted, and how that information is returned. I do think that in terms of the culture, it has the potential to change both the culture as well as the infrastructure for being able to do that, such that there's going to be a different expectation as well from patients as participants about what they're expecting, and they will vote with their feet in terms of what studies they participate in. So I think just those in terms of policies as well as ways of being able to do that.
I would just comment, I agree with the comments made, but I would also bring up that many clinical trials are global in nature, and so we do have to respect the laws and regulations in other countries as well. So we would want some sort of, compatibility or harmonization, and also recognizing that in a global setting, the support that is necessary may not be present, and there may be cultural differences, and that would need to be taken into account as to whether the same approach would be used globally or there would be differences that were accepted as a responsible way to do it.
Yes, I think there's another piece of infrastructure that we need that possibly the government could set up, and that is a database. The onus would be on the patients who had participated in the trials to keep the information updated, but it would be up-to-date information on contact information basically on the patients who had participated in clinical trials. When I was at the NCI, we had funded big, useful clinical trials that even if you wanted to get in touch with the patients afterward, you could not because you couldn't find them. And it was just cost prohibitive or even impossible to track them down. So if there were a way that, that this information could be centralized and made available so that patients could be contacted, that would be an enormous step forward.
So can I follow up with Jessica on this question? You said policy and legal changes. Did you have specific things in mind?
So when I think about that, I think about really how complicated it has become for us to return genetic results across all 50 states using a centralized bank of genetic counselors. Because of some states have very complicated licensure laws that actually are prohibitive for a genetic counselor based in another state to get licensed there. And so as we think about scalability, there are not enough genetic counselors out there to start returning results. So is there a way to remove some of the barriers that are in place for these, that are with state laws? And then the policies, I get back to thinking about the limitations of GINA. And what protections are not in place. So small employers, I think about an All of Us participant who's a young individual still working, but working for a small employer, learns their genomic results that now their employer can discriminate against them, right? GINA doesn't have all those protections, nor does GINA protect against return of some biomarker results, like I was talking about the elevated brain amyloid. There's no protections there. So those are just two examples. I'm sure there's plenty more I could come up with.
Thank you very much. Chester, Wiley, and Jeff, I have you also.
I think a pretty straightforward question in that, Wendy, you mentioned the possibility of returning BAM files or some other sequencing format directly to subjects and letting them kind of work it out. Do you have a sense of the downstream model for that analysis? There are concerns about quality control for bioinformatics, et cetera, and how that might be contended with, perhaps in a similar model that you suggested for, you know, just not everyone becoming Clia certified, but having centers that did something like that.
So I agree that it is potentially a solution. The model that, and I'm not proposing that necessarily Illumina be the one to do this, but Illumina has a Grail model now, in terms of being able to have apps where you can have a subscription to being able to have your genome interpreted for, condition X, Y, or Z. Right now, and I'm not suggesting that the FDA necessarily be the ones to regulate that, but this is relatively unregulated at this point. I would love to see something where the community actually got together and gave it sort of a 5-star rate, scale of 1 to 5, the ratings from a community-based point of view. What do the experts think about this in terms of how accurately this information is being interpreted, being able to have those out there. The nice thing about from the locus of control in that is that the patient is in control then, right? It's their data. They can decide if and when and how they unlock those, open those envelopes and find out that information. But I do think what you're pointing out is right now the limitation is that either the interpretive accuracy in terms of a lot of that information is what's in question. And I actually, frankly, have to say that that's true of all genetic testing on the market right now. It's not just simply interpreting your genome if we give you your BAM file.
Wiley?
I want to get back to the issue of Clia certification. I think what I'm hearing is it's a contributor to quality assurance. It's not the only aspect of quality assurance, but it is an important contributor. And I think we're also hearing a consensus that not all research laboratories realistically should pursue Clia certification. So I'm trying to figure out how we should be thinking about the role of Clia certification in return of results. Would it be reasonable to say that it's optimal for a research result that goes back to an individual participant to be confirmed or from a Clia certified lab, but that one could imagine circumstances where a result might go back that's not from a Clia certified lab? Or should we be looking at an absolute rule? could you contemplate circumstances where there's a result, it's not from CLIA certified lab, there isn't a feasible pathway to CLIA confirmation, but yet the result should go back to the participant?
So I'll just give a couple use cases to think through this in specific terms. So we've had circumstances in which we've had, through something called the Breast Cancer Family Registry, an NCI-funded study for the last 25 years, women with breast cancer who have passed on. And the only sample that was available was a sample that came in. It is not possible to get a new sample to go in CLIA certified space. It wasn't in CLIA certified space. We can't return that. But yet there have been descendants of that woman with breast cancer who have wondered about the BRCA status. And so we have gone back to our IRB in certain circumstances of that and been able to say that we have taken that sample. That sample was broken in terms of chain of custody, but at least we did the clinical confirmation in a CLIA certified laboratory, so we got as close as we could to in terms of doing that. And we're just very careful in terms of explaining to the family members what it is that we found out. And that was done in the service of being able to interpret their genetic results, which their new genetic results for descendants were done completely in CLIA space with chain of custody and everything else. The other thing that we've done in certain circumstances is we've had samples, as I mentioned, we're doing a very large genomic study where we take the sample, it comes in and is processed in CLIA space. The exome sequencing is done in research space, and anything that's reported out to participants is confirmed with Sanger sequencing of that original sample still in CLIA space. So it's sort of a hybrid model that we've done to try and scale this while still being able to make sure that we've got the highest quality possible for participants and doing it on scale.
Well, we did this all the time in CLGB over the 15 years that I was there. So the correlative science research laboratories that use the specimens that came from the patients on the trials were not CLIA certified. But if there was a clinically important finding or an unanticipated finding that had nothing to do with the trial or the disease being studied on the trial, but something completely different but important, we did return the results, but not to the patient. They went back to the treating physician who had put the patient on the trial, and then those were followed up by testing in a CLIA laboratory to see if they were reproducible and indeed verified.
I would just say that, CLIA does not need to be the standard, but there does need to be some standard. Right now, research use only tests, don't meet a recognized standard. And in order to ensure accuracy and have that sort of confidence in returning results, there would need to be some standard recognized that could be applied.
I agree, especially for something that could be confirmed in a CLIA lab. I think then there's no reason why you would need to do it in a CLIA lab. For instance, we know that there's genetic assay that people do assays in research labs for testing outside of CLIA. The results are not always accurate, and we see this time and time again for APOE for Alzheimer's disease, reporting inaccurate results when they're done outside of a CLIA lab, and those have significant implications on the individual they're being returned to.
So can I extend that question a bit, which is it realistic to have every result? When you talk about a genetic result, it's one thing because you can do a lot of kinds of sequencing in a CLIA lab that does genetic testing. But when you're talking about other kinds of biomarkers, you can't just go repeating things in a CLIA lab. So you kind of each of you pushed back to say, yes, it has to be done in a CLIA lab or some hybrid model or something like that. That's fine maybe for sequencing, but CLIA doesn't allow you to just do a test off the cuff. So what's the reality of repeating a research result in a CLIA laboratory before it's returned? Is that realistic?
Most of the tests that are done in drug development are exploratory in nature. They are, they're really not that useful when an individual data point is examined. It's when you look in the aggregate. For example, perhaps looking for new drug targets in a disease, and there may be an overall association when you compare groups of people who do and don't have the disease, or looking at potential biomarkers of whether a drug is perhaps perturbing a biological pathway through which the drug is supposed to be working. So the value of returning those is very questionable. And to develop a Clia test for that, in a timely fashion is really not realistic.
Right. I think my comments were concerning purely genetic results. So in our field, we think about brain imaging data as our biomarkers, and there's no Clia lab test for the PET test or MRI, and then the CSF assays are rapidly evolving all the time. So I think there's significant limitations about a blanket statement of Clia regulations for return of research results.
Thank you. I have Jeff, Paul, and then Stephen. So Jeff.
So my question is primarily for Leah, and I think industry is a particularly important stakeholder in this domain because you fund and design your protocols. And so what you've described to us is a lot of the testing of biospecimens in that context is exploratory now. The tradition is typically not to return those results. So I guess I'm wondering how considered that stance is at this point. Is that basically just a tradition, or has pharma taken this issue up and is it being evaluated on a protocol by protocol basis? You're part of the industry pharmacogenomics working group. Has that working group taken a considered position on this? So where is pharma in this debate right now?
So the Industry Pharmacogenomics Working Group is among many stakeholders that has considered this. In fact, we put out 2 papers on the considerations of return of research results. It is an area of active discussion within the pharmaceutical company, within individual companies. It is considered on a trial-by-trial basis on what the appropriate thing to do would be given what the test is, et cetera. As I said, most tests don't meet the threshold of being clinically actionable and Clia certified, so it doesn't come up as often as one might think. But yes, it is being considered quite seriously.
Paul?
So back to this Clia issue, there's an assumption, and some of you have stated it explicitly, that the results coming out of Clia certified labs are more accurate. And I'm wondering if we have any data on how much more accurate. That is, for those of you who have had experiences taking research lab data and then having them Clia confirmed, how often don't they confirm? And I understand that may vary by the type of test that's being done or exactly what you're looking at. But are there any data at all to suggest the dimensions of the difference?
I'll say not all research labs and not all clinical labs are the same. So just start by saying that. So I have had circumstances where we've taken research results and then done clinical confirmations on them. And just to give you a general, as a QC, there are times when we can figure out sample switches. So just as a very simple thing, as a simple mistake that can be made. Sample switches in my research laboratory, I'll air my dirty laundry, happen about 1 in 500 times. I don't know where that is, if it's at the points of blood drawn, saliva contribution, whatever it is, but things where I can see a gender swap or some family relationship that doesn't make sense, that I can say something happened somewhere. With the clinical labs, when that happens, in terms of doing that, there are a lot of things that I can say, at least we did in my clinical lab, putting in molecular barcodes so that at least we knew that from the time point that it hit the laboratory, we did not have any sample switches, at least from the robust processes that we had in the lab. And we had a lot more automation in place to make sure that we didn't have human hands in terms of sample switches. So I can say that in the few circumstances that it had, we always caught it before it went out the door. So I can't say that there weren't ever sample switches. about 1 in 5,000 times something would go wrong, but we always caught it before we actually reported it out as a result and then did a root cause analysis to trace back where it was. So at least if we were having that, I would say it's an order of magnitude difference, at least with very, very robust, large clinical labs that are doing this. But not all clinical labs are necessarily as large or as robust in terms of the QC that they put in place. And that's just for molecular genetic testing.
Steve?
So I wanted to follow up on the Clia discussion, following on Wiley's question. Unless I missed it, and if so, I apologize, there was an ambiguity in the responses. Certainly the model of the result is done in a non-Clia environment, but confirmed in a Clia environment. I think there was sort of broad comfort with that on the panel. But what I don't think you addressed was an alternative model, which is a result is, or test is done in a non-Clia environment. That result is returned to the participant or the appropriate person with the disclaimer that this was done in a non-Clia environment and no action should be taken on it unless and until it's confirmed in a clinical laboratory. And I just wanted to get your responses to that alternative model of invoking Clia.
I think in my experience, if patients were to go that next step and do the clinical confirmation on their own, that would be fine. In my experience, patients say, we trust you, you're researchers, we bow down to you, we believe you, and they actually don't go the next step in terms of doing the Clia confirmation. And in rare circumstances where I've had women, for instance, that are considering bilateral mastectomy for something extreme, even in a clinical lab, they will get a second opinion and get that result confirmed because they really want to make sure that it's the right decision they're making. So, but short of very, very important sort of high stakes decisions, I just, in practical experience, I haven't seen people do that.
Rhonda, and then I would like to see if there are any questions from committee members on the web.
This is a very basic question. So, Wendy, you talked about, you obviously have a Clia lab and a non-Clia lab, and you mentioned that there's quite a range in labs. Is there a basic standard that every research lab must adhere to, short of Clia? Is there some reference standard that labs are expected to adhere to, or is it completely open to the lab head how procedures are carried out?
To be clear, this is research laboratories or clinical laboratories?
Research laboratories.
So research laboratories, it's a free-for-all, right? I mean, people are doing lots of different things, lots of different methods. There's protocols that people follow, but there are no standards we don't go through, and we don't do the same thing we do in a Clia or a CLAP environment in terms of doing certification or being able to do validation studies or anything like that. So it's meant to be all over the place because it's research. I mean, we're exploring, and many of these, as we've talked about, many of these methods are just in the development stage. I mean, far less of standardization, and that does contribute to the reproducibility problems, and in many cases, they're not meant to be used clinically, so there's no need necessarily for that rigor. So, I don't think there's a problem with that. It's just that that's not the way we run research laboratories in general.
So can we, if there are committee members on the phone, if you have any questions, now's the time. Nope. Richard, and then Jeff again.
We heard earlier that some people would like to have all of their data. I haven't heard anybody talk about if people wanted all of their data, say from genetics, what would that entail? What, on the part of the researcher to provide those data? How big of a task is it? Is it realistic? Is it worthy? Is there some way to circumscribe all of their data to being not all of their data? How could that be handled?
So I'll deal with this just from the genomic aspect, and so we do a lot of exome, genome, transcriptome sequencing. We don't advertise, we don't try and sort of push out all of their data to everyone, but if a participant will ask me and say they want their BAM files, I will give them a jump drive with their BAM files, and we actually organize our files. We have the infrastructure so that I can do that without pulling my hair out to be able to do that. It is not that difficult to do that. The difficult portion for me is to actually do the interpretation. So if you say, I don't just want that, but I want to understand what is my breast cancer risk, what is my Alzheimer's risk, go through that. That's a difficult process, and that's incredibly time intensive. So a very simple solution would be to say, okay, at least on the genomic side, here are your data, you're now in control, you've got the power to do it, what you do with it is up to you. I have had, and certain of us, even in this room, have had discussions, for instance, should we not just give a BAM file? A BAM file to most individuals, honestly, they'll put it in a drawer and they won't do anything with it. I mean, they feel better that they have it in case they might want to do something with it someday, but they don't know how to use it. They don't even know how to open the file. So other people have said, well, would you give people a VCF file? In other words, a file of variants that individuals could then sort of play around with and be able to understand. I actually think that's potentially dangerous in the sense that a lot of lay people don't understand what that information means and can jump to conclusions about risks of all sorts of different diseases because they'll see a gene listed, they'll see a particular variant listed, and truth be told, even if you go into ClinVar right now, we're not as a community at consensus about what a lot of variants mean. We don't understand a lot of that even amongst professionals. So I think it's, in my opinion, more dangerous to be able to give a long list of variants VCF files like that than to be able to give someone the raw data from which they can start from ground zero and do whatever they need to. The biggest problem I have with that is just I mentioned it before, it's a disparity question, right? So not everyone is going to have the access, the resources to be able to pay, I don't know what the fee is going to be, but let's just say it's $1,000 to have your genome interpreted. And so, is there any wedge that we're driving in here in terms of disparities in terms of those research participants? That's the main disadvantage I see to that model.
Do you know what the services that you describe charge for an interpretation of a genome?
So I don't, and someone else can certainly answer this, I don't know of anyone right now that is, I shouldn't say that. There are certain services that I imagine the price is probably running in the neighborhood of $1,000. I don't think that's far off. I think what I've seen more commonly is services that are trying to interpret a subset of the genome on a gene by gene or disease by disease basis. And, those run more in the couple hundred dollars, that sort of range.
Jeff, do you have other written questions?
I do. This is from Consuelo. How do we reconcile some participant perspectives that they co-own data versus researcher perspectives that biospecimens are donated and volunteers relinquish ownership?
Well, I will say that over the last 5 years, I deal with a lot of families that have rare genetic diseases. And whether it comes to, I've just seen a lot of, I think in a positive way, active engagement from those participants. And they are demanding now much more in terms of ownership and having a stake in everything that goes forward, whether it's their actual specimens, whether it's their data, whether it's in some cases cell lines that are derived from them, iPS cell lines, other things that are coming from them. And whether it's usually not an individual participant, but it's as a group of them, a nonprofit organization, for instance, that is collectively saying, we are owning these, and therefore, largely, I think in a good way, to be able to say, we're going to make sure they're freely accessible. We want to make sure that we're removing the barriers and that everyone can be able to access this on a level playing field. And I don't think they're so much worried about making money from it as just making sure that the most good can be done from those things that come. I know it was said many times before, but I think it's all in terms of you just have an agreement up front with the consent. I think it needs to be very clear, very transparent, very understandable in terms of this. I think realistically it's very hard to implement in terms of individual patients continuing to have ownership down the road. I mean, just being able to track that and be able to get permissions, I just logistically have trouble in my own mind figuring out how to do that. So I think the model is still easiest to implement and probably truly what people are trying to do, to donate their specimens, to realize that they are relinquishing that. That's open and agreed upon at the very beginning for that, but that there is certain information derived from those samples that, if individually useful, could be going back. Practically, that's to me what makes the most sense.
I would just add a couple points from the clinical trial perspective. I certainly understand both perspectives about who owns the data. One situation which could be problematic is if the ownership were solely on the part of the participant and they decided they wanted to remove their data from a database, the clinical trial database. That would compromise the integrity of the study. So that sort of action would not be a good thing. I would say that in terms of if ownership is thought of as controlling what's done with the biospecimen, that is all set out in an informed consent form. Even for our samples that are used for future use, it may be shared with other investigators. Since we anticipate that may be done with the samples, we do outline in the consent form what the parameters of that research will be. And if someone were to desire to do research outside of those parameters, additional IRB review would be sought, et cetera.
So I have Chester and then Greg. And
back to the question of Clia requirements for research laboratories. In thinking about this, just the potential gradient here. Clearly, the intervention in the case of breast cancer identification or gene susceptibility to this is quite high. It's a very high-risk result, if you will. But in rare disorders, let's say, give the example of a clearly pathogenic variant that was derived from a research study. That information was conveyed to a clinician who understood that and knew the clinical phenotype of the patient and thought that it was a good fit, completing the triangle, if you would. Could one consider kind of a gradient model in which the potential impact of intervention is thought about, the clinical input in linking that research result to the patient's phenotype could in fact alter the Clia requirement? Is that something that the panel feels might be a reasonable thing to do, or should it be an all or none situation, I guess, irrespective of what the condition is that's being considered?
I'll just start off with more of a question back in the way that, what if this person is asymptomatic? So there is no clinical phenotype yet, but they're in for something else. And then you're informing on potential future manifestation and thinking about the psychological impact. So I realize I'm not answering your question. I'm just adding another level of complexity, perhaps, to that gradient. But it's a very good question and one that we should consider.
I think they're two separate things, and the Clia certification is important. So the clinical aspect can certainly address what does this mean for you, what might you do about it, but the Clia certification really gets at the laboratory component of generating that result, and they are distinct things that those two sectors would cover.
I think what needs to be kept in mind here, though, is the test itself that's being used to generate the data that implicates a problem for the patient. That may be done in a non-Clia environment and give you a correct answer. And it can be done in a Clia environment and give you an incorrect answer because Clia doesn't regulate the test itself. It's the environment in which the test is done. So I think that some focus needs to be placed on, how confident you are in your analysis method, in the technology and the methodology being used in any environment for this kind of question to be asked. So I just wanted to add that little footnote to Clia.
Greg?
So the former FDA Commissioner Rob Califf talked about small, crappy trials like last year, and NIH has put new parameters in place to sort of improve the quality of clinical research through NIH and really sort of beefed it up. We talk about, I'm not a laboratorian, so I'm trying to understand. So we have CLIA labs, then we have research labs, but there has to be a spectrum there of small, crappy labs, and then these big clinical trial networks that have laboratory cores that have been running for 20 years or even more, which have superb quality control, and you could see a greater level of confidence in these larger laboratory cores that are part of clinical trials networks than sort of a laboratory at my institution that does a clinical trial related laboratory experiment every few years. So it seems like there's research labs are not all created equal in terms of and is there any way to sort of, as we're thinking of returning research results, figuring out a way to improve standardization of the non-CLIA labs?
I just think that effectively, if you end up doing that, you end up putting in place basically what end up being pretty close to CLIA requirements in terms of doing that. And again, it is the environment that you're talking about in terms of the laboratory. I would love to see, in addition to that, the rigor of the reproducibility and the validation of the individual tests if you're going to return that. And we do those within CAP certification right now. So that would be my ideal. But effectively, you've turned those research laboratories that are those big monstrosity, really good, high quality, you've essentially just turned them into CLIA labs. So, and I would be supportive of that.
Jeff?
I guess my question sort of builds on Greg's to some extent, and it seems like all the discussion about CLIA focuses on the individual lab, and what does that lab have to do in order to get up to speed? What's the role of research institutions like the University of Utah? If we wanted to say we want to meet this expectation of higher quality and return of results as an institution, is there a way that academic institutions can work to foster CLIA certification for a suite of labs within that institution, or is it almost always going to be a lab-by-lab project?
I mean, I haven't done an inventory about what's on the table for return, and I'm a genomicist, so think solely in that dimension. To me, it's about then thinking about as institutions or even organizations of institutions to be able to consolidate that, right? So in that particular case, there's an economy of scale anyway in terms of just producing good research data, driving down the cost of testing, being able to do things on scale. And so I would actually say that as institutions, you're better off rather than having every single laboratory doing this, trying to have a similar workflow, similar data generation interpretation that builds in the rigor for that, and being able to have, at Columbia, we would call these cores, but either cores, genome centers, things like that, at least when it comes around the genomics. And for that, then you've got the best, to me, the best of both worlds in terms of being able to do this on scale and still have some local control.
So could you speak to the aspect of research laboratories, which isn't really being considered here, is that they're training sites. They're scientific training locations where you have graduate students and postdocs who aren't necessarily competent in the testing that they're doing. And how does that play into research laboratories returning results to individual research participants?
I think this is incredibly important, the proficiency of the people doing the tests. Everything else can be perfect, and you can actually generate bad data from someone who is not competent in doing the right thing. And I also worry, I hate to bring this up, but it's related to the rigor and reproducibility problem. The amount of how can I put this, made up data in research, it is a problem. And there is an organization called Research Watch, I think, that is actually monitoring this. And so I would, the standards in the research laboratory are really different. And it's unless you absolutely know through licensure and through proficiency testing the competency and the reliability of your operators, you also can't count on the reliability of the end result. So I would vote for a strong yes for that.
So I have Steve.
So actually I have two questions if nobody else has their card up. So the first is, implicitly and at times explicitly, various among you have suggested that there's a need for clinical sort of counseling to accompany the return of results. And so again, one model might be that the research lab somehow is working with genetic counselors or other clinicians who are licensed and skilled to do this, and that's part of the package of the return. Another model might be that you rely on the clinicians, the primary care physician, genetic counselors that the patient might otherwise have access to through their sort of clinical care to fulfill that function. What's your view about the responsibility of labs if they're going to be returning results to have the genetic counseling or imaging counseling or whatever other type of counseling be a part of the package that the lab offers?
So, as I was describing, we had started this study Spark for autism and returning the research results, and I wanted very much to be able to have those returned results to those participants embedded within their medical home. I wanted them to be actually returned by their primary care doctor, their own medical team, and we actually had clinical sites in place, we thought, to be able to facilitate that. And so my original vision for doing this was exactly that. We would hand whether it a pediatrician, neurologist, psychologist, whoever, this information. And I got the strongest pushback I have ever gotten because people said to me, I have no idea what this is. I don't know what it means. I look like an idiot trying to explain this information. I did not order this test. I do not want this test. Take it away. And so that was when we decided that we needed to actually have something in place, that we still have the option for people to be able to get it returned by their designated provider locally, but we set up the opportunity, and the way we got around the state licensure issue is we were actually working with an organization that has genetic counselors that are licensed in all 50 states. I didn't personally have staff to be able to do that, so we needed to go through a for-profit company to do this, and so that's the way we're doing it. We are still, I have to admit, relatively early days. It's new to me in terms of how to be able to do that. I would love to be able to do this with video counseling and be able to get body language and everything else that you get. We found that participants just couldn't technically get that up to sort of be able to do everything they needed to do. So it's a telephone disclosure in this with a video prepped ahead of time so participants can sort of have quality control in terms of a standard message that they're getting, and then tailored information from the genetic counselor for their particular genetic result and their particular family. So far, based on the feedback we've gotten, it's working relatively well. It's not, I don't think, will ever be as good as face-to-face from your own particular genetics team, but it seems to be going well enough, I would say.
That, of course, is one model where at least part of the purpose of the study is to return results, and so you're going to it makes sense to do that in the context of that study, but there's a huge diversity of situations where the question of return of results might come up. And what I'm wondering is, do labs have to be thinking about having a genetic counselor either in-house or that they're working with somehow to have this as part of the package of services, or else they shouldn't be returning results in the first place?
I mean, for our study, it is not explicitly studying the return of results process, and so this was just thought to be responsible in terms of how you get this information back. I am worried about just sending an email to someone in terms of this information and assuming they can get that and find their way around on their own. So at least I'm advocating for you need to do it responsibly for many types of results. All, I don't know, but many.
And just to build off of what Wendy was saying, I think that you can envision either model working, right? But it's also giving the right communication tools either to the the primary care doctor to be able to communicate these potentially very complex results that they're not trained or skilled in, and thinking about what the disclosure materials look like that are written in an accessible language, are clear, concise, and have appropriate conversations. I don't know that labs are equipped to be able to offer this, and it's an interesting idea. However, again, what are the disclosure materials that are available for a wide array of results? I don't think we're prepared to even talk about some of what's these variants and what they mean in terms of a person's risk for a particular disease.
Steve, did you have a second question? Oh, Rhonda?
Well, mine sort of builds on Steve's. So Wendy, it sounds like you outsourced some of it, but this isn't a core that serves other labs. So could you comment on the feasibility of, since some labs will not have any clinical expertise and some clinicians will not have any knowledge of the research result, is there a place for sort of a third party, maybe developed by the institution, that has the expertise in the return of results.
So that's a great point. I guess that's the point that I was trying to make, but not so clearly, is that I think there's the opportunity in terms of being able to let research laboratories focus on what they're good at focusing on and being able to do this return of results in modules that can be outsourced. So one of those things to outsource is, I think, the actual returning of the results in terms of who presents that information. I think that even though, so we outsourced that to a company to be able to do this. This wasn't the laboratory that did the confirmations. It wasn't the research laboratory. This was an individual module in addition to that. I think the question is, how do you, if that's the forecast of what the community is going to be doing, we will need to build that infrastructure in terms of other modules to be able to do those pieces. I will say, as we were doing this, in terms of being able to allow providers to return this, I did everything I could. We made simple patient-friendly brochures about each result that was given, videotapes to do this. I sort of prepped them ahead of time to be able to do that the best way I thought they could, and I still got a lot of pushback. And I wish it were different, and I wish genomics were sort of not so scary to so many providers on the front lines, but it still feels like it is. And it's time for them. They're not being compensated for that. The incentives aren't aligned appropriately. And so, I just think we just have to build this into the forecast from an infrastructure point of view as well as from a budgetary point of view.
And I can say we've also, quote unquote, outsourced our return of results for one of our clinical trials, but through an academic group at the University of Pennsylvania. So there are models, but they don't have the bandwidth necessarily or ability to get licensed in, I'll say, the state of Hawaii, which represented a very complicated legal landscape. So there were limitations, but there are opportunities and I think which are important for scalability of return of results, to think about creating centralized banks of licensed trained providers who can think about how to create the materials that are written in a way that can communicate these results in an accessible, clear way for diverse populations.
So my other question is whether any of you has any concerns or experiences or knows of any experiences with legal liability for research labs returning results.
You're getting blanks, so
I'll
I
take
think
that
the
as
answer is
no.
No.
No concerns? Maybe no experiences or no that you're aware of, but I'd be surprised if there are no concerns about this issue.
I mean, the concerns to me they haven't come up on the research side, mainly because we've put that off on the clinical labs to do the interpretation. So the liability issue to me has been pushed off. It's been outsourced in that way. It's been pushed off to the clinical labs. And there are the usual liabilities that we have on the clinical lab side, which is that when you screwed something up, right? But I think we've tried to minimize that risk, but we've simply pushed it off to the Clia labs.
Yeah, I don't know what the liabilities are, but I think they're there, and perhaps if the research lab returned an incorrect result to the participant, how is that handled, and whether the cost, could they bear those costs if a lawsuit came up?
Vanessa?
Wendy, I'm interested to hear more about this pushback you got when you said that you wrote, a scenario in language that you thought people could understand. What was, the nature of it? And have other people experienced that? And then when you had to outsource it, who paid for that?
So the nature of this was so it wasn't universal. It wasn't 100% of people that didn't feel themselves competent to be able to return results. The number is running basically about 10% of the providers that are approached feel competent, feel completely comfortable, or have someone on their team to do this. So it's not a universal thing. The folks that I was surprised by, maybe I shouldn't have been, but were in this particular case primarily primary care providers in terms of being, again, my sort of naive way of thinking about this was that should be the medical home. That should be that someone feels very comfortable with and is in the setting where it's going to then integrate into their clinical care, and that would be the appropriate person to be able to deliver that information. And I was just dead wrong on that in terms of their level of comfort with that. Over time, I have this sort of, naive optimism that over time, a lot of the problems we're talking about are going to get easier because participants, physicians, providers are all going to get more sort of conversant at this. It's going to be easier to interpret the genome and to understand the genome. So I think this is going to be, I hope, a downhill battle at some point, but there's going to be this time when it still continues to be a lot of pushback for me in terms of this. So in terms of the question of cost, I have to full disclosure on this. This is a study that's completely being funded by a private foundation, so I don't have to worry in terms of NIH about fitting this into the budget and taking money that I allocated here and then stealing it away for something else, or worrying about a 20% cut in my budget or things like that. So this was, but it was with forethought that we knew that this was from day one, because we engaged participants, we knew this had to be a part of the study design, and we knew that we needed to allocate resources for this. So, you know, it ended up changing buckets a little bit from, you know, this source to this source, but we had, you know, return of results at front and center in terms of knowing that we would have to put money into this.
Right?
Hi, this question is for Wendy and others with experience. So we just heard you speak about the provider perspective on using the medical home as far as returning results. Do you have experience with participants saying they didn't want their results returned to that model or linked into their health record or medical home?
So I've definitely had that experience, and we've had several different types of research studies. Some in this room will know about eMERGE as an example, in which it's very much tied to being in the medical record, and we certainly have had many people say just flat from the beginning, I don't want to be part of that study because I don't want this going into my EMR. And so different studies have certainly said they did or didn't want that information into their EMR. There are certain individuals who do or don't want specific providers for whatever reason to have access to certain information. We have tried to remain with things in the locus of the control of the participant by trying to be very transparent about what are the upsides and downsides to having that information in a place where all can see. I would say the vast, vast majority of participants, when there are things that are medically actionable, absolutely want that integrated front and center, very clear on their problem list, want everyone to know so that they can help them take care of themselves. So I don't think there's been so much of the, you know, conspiracy about that. You know, what we heard about in terms of still concerns about GINA and issues about long-term care, disability, life insurance, they're still there. They're still concerns, and that's the pushback that we get. Interestingly enough, I get more pushback in terms of individual results from physician providers being concerned about that than I do from regular off-the-street patients. To a certain extent, that's understandable. They're worried that their home is also their work environment and discrimination from their colleagues in terms of actually knowing that information. But I would say, you know, 99% of people put it integrated into their EMR. There's not a huge amount of pushback about that.
Jeff?
Yes, question from Amy McGuire.
I think Jeff's got a comment.
Oh, I'm sorry.
Oh, I was just going to say, I think it probably depends. So for APOE, for risk of Alzheimer's disease, there is still persistent fear about it being added into the medical record for fear of, because it's just a susceptibility factor. It increases your risk, but it doesn't say with 100% certainty. So there's many, many people who say, I don't want that part of my EMR, you know, and so they don't want it to go back to their treating physician, et cetera.
Yeah, so this is similar to Bray's about choices. So this is from Amy. Do any of the panelists have experience with offering participants options about receiving results, and is that feasible? One panelist mentioned that some participants don't want this information. How can we best manage differences in preferences for receiving results?
So when we've done this on scale, it's been bimodal. We always offer preferences, but when we do this on very large scale, it's either yay, nay. I want my results that are related to the primary indication of why I'm in the study, and then we've offered the option of yay, nay, I want secondary findings or incidental findings. We haven't gone to that level of granularity when we've done studies on very large scale. When we've specifically done studies to look at participant preferences, we have offered all shades of white, black, and everything in between, and that is just not scalable. It's very interesting to study and very interesting to understand what preferences people have and why they have them for certain information, but it is just not feasible to do on scale. So you have to, I think, have some probably dichotomous, you know, again, we've done it as main reason for the study, and then secondary or incidental or actionable findings, however you want to word that, knowing that that may change over time as medical science changes.
One follow-up to that. Oh, sorry.
No, I would just add that I agree that it would be require considerable infrastructure and cost. In my own company's consent form, we have a tiered consent, not about returning results, but subjects do have a choice. To keep track of that information and have a system in place for that involves training investigative sites to record the information correctly, get that information through a data management system, get it cleaned, be able to access it. It's a considerable infrastructure to do that and to get it right, and I just wouldn't want to underestimate that.
Yeah, and follow up perhaps primarily for Wendy. I don't know whether the studies you're referring to are hypothetical attitude studies about results, and whether you see a distinct difference between how people think they want results and how people actually request results when they're available.
So we've done that with our, literally it was called our return of results study from exome sequencing. So we took at baseline their preferences. We also asked them then after we did a formal genetic counseling session, and you'll hear from Julia Wynn tomorrow, one of the genetic counselors on that study, when we went through the down and dirty of exactly what does it mean. Those counseling sessions were oftentimes 60 minutes or longer in terms of doing that. And we did see probably about 20% of individuals change some one of their preferences, usually not an all or none, but some nuances in terms of specific things. So yes, I do think the more people are informed, the more they understand the granularity of this, those things change. But I will say that most people, when it comes to, at least the ACMG secondary findings list, it's pretty obvious to most people that they want that information. If there's something that could save my life or save the life of someone in my family, most people want that. In my particular community, I have some individuals, for instance, who are Orthodox Jewish, who have, it's a scarlet letter G on their forehead if they have anything genetic. They don't want that information, and I think that's perfectly reasonable for them to make that informed decision about that. So I think in general, that's sort of the workflow that we've built into things to scale them.
So I'd like to offer the audience or the people who came to the public session, if you have any questions, to ask the panelists. Yes. Is there a microphone to use? Yeah, there's a microphone coming. And I would ask that it be a question, not a commentary, please.
Thank you. I'm intrigued by the difference between the desire of patients who want the information and then the sort of the pushback we'll get from clinicians and others who feel the responsibility of giving that back in a way that would be useful for the patients. I'm starting to wonder, and Wendy, you sort of talked a little bit about, as people get more familiar with this, and that's across the spectrum, researchers, clinicians, patients as well, do you see a peer-to-peer model? Do you see an opportunity where patients and participants themselves begin to get more engaged in understanding what this means and then can pass that along or pass that forward to others like themselves? I should say I'm Sally from Patients Like Me, so this is logical.
Yeah. So we've actually started doing that in the sense that, and we do this through the form of stories. So we actually take individuals who have received their results back and allow them to tell their stories on video, sort of a StoryCorps type of thing, which is a very relatable sense then to other people because they know they've been there, they've done that, and it also then doesn't require every participant to be a big brother, big sister to someone else. It's scalable in terms of thinking about that. So that has been helpful. I think we're still in the process of getting enough stories to get enough perspectives and point of view because everyone's different in terms of this, but I do think that's part of what is going to be eventually the level of comfort to scaling this. Just as an example, and I know many of the geneticists know this, I mean, we saw a tremendous difference after I call it the Angelina Jolie effect, right? So after there was a lot within public discourse in terms of what Angelina Jolie had done in terms of her BRCA status, people they read more, they talked about it more, they got comfortable with the issues, and I think this naturally will evolve as we're talking about other things. It's going to take time, and the more we can do in terms of public education as well as provider education, the better we're going to be.
Going once, going twice, lunchtime. I'd like to thank all the panelists and the committee members for your participation and the audience, and thank you very much.
Just one housekeeping thing before people go to lunch. We do have lunch right outside for the committee and for speakers. For the public, there is a cafeteria downstairs, and both Olivia and Caroline right there will help direct you to it. And we'll meet back here in a half an hour at 12:30. Thank you.